compounds. So they all had to be drug-like. And so at the end of that, we built up a portfolio of compounds. And we've got one lead asset that were good enough that we decided to spin out a company. And that was Alevin Therapeutics. I think it was long enough, isn't it? It's still not changing. I'll continue. We incorporated in 2022 with seed funding from O2 Ventures and University of Nottingham. We got about $1 million. Sorry, $1 million, yeah. And so we've spent the last 18 months basically advancing our lead compound through to a position where we're in the next stage, the major stage now, of taking it into safety studies. And that's the kind of story that I'm hoping to tell you this afternoon. Okay.
I mean, it's a heavy file, maybe it takes a while to move. So
Yeah.
maybe just leave it for a second. We'll give you extra minutes, Tom, don't worry.
So anyway, so our lead compound is 296. And the difference between ourselves and our major competitors, or a major competitor, they're an oral compound. So we're going for an inhaled approach. So that's why I've underlined inhaled. Yeah, so here we are, 2022, million seed funding from Sunil plus University. And basically we've taken our lead compound, and it was a 6 nanomolar, so it's very potent. We showed it was efficacious in an animal model, which is the standard global model. But more importantly, to get it through to the next stage, we've done 14-day repeat safety studies in rat, proof of concept in, say, animal model, and importantly, in human IPF tissue. So this is tissue from a lung transplant, and I'll explain that as we go through. We've done some solid state stuff. Yeah, it's a crystalline form that's developable. So we've got a development plan in place. But importantly, with our pipeline, we want to have a pipeline with other fibrotic diseases also. So we're currently looking at some compounds in various cancer cell lines, and it's looking pretty good in ovarian cancer cell lines. And we've got some backup compounds. Being a farmer, I was always told, get backup compounds because things fall out in development. So we'd like to get backups to basically add value and de-risk the program. Thanks, Neil. So this is our pipeline, 296. It's in the preclinical phase. IPF for inhaled. It's our backup compounds, and they're currently undergoing some DMPK, and I'll show you some data later. This is an oral series of compounds that we had in the university, and we've proven that these are orally active, so that we might go switching in the future to an oral series also, and we've got various other things for cancer and AKI and other kidney diseases. And at the bottom, you can't really read it, but basically, one of the other big fibrotic diseases, obviously, is NASH, and it's now called MASH. And it's the same mechanism. Okay. So it's going to. So IPF itself, there's this statement from this paper said 45% of deaths in the Western world in some way implicated with fibrotic diseases. Fibrotic disease, what is it? Well, fibrosis is deposition of extracellular matrix, scar tissue. IPF progressive, and unfortunately, it's a very poor prognosis from diagnosis to death between 3 and 5 years. Quite a number of people worldwide, and with an aging population, the numbers are increasing. In bold here, I've said no effective treatment capable of halting disease at the cellular level, and that's true. The two compounds on the market, there's Boehringheims nintedanib and pirfenidone. Revenue you can see here in massive revenue, but unfortunately, more than 30% of the people come off those treatments because the side effects are dreadful. Nintedanib primarily it's GI, severe diarrhea, and with pirfenidone it's more photosensitivity plus GI. Very large market, and we believe that what we want is a disease-modifying treatment. These are symptom disease treatment. We want a disease-modifying treatment.
Yeah.
You get it? Okay. So the picture on your right is a standard IPF patient. As the disease progresses, the major treatment is supplemental oxygen. There's some stuff there. But not everyone holds them back. This is a Scots guy, 82-year-old cricketer, and there's his oxygen cylinder on his back. So yes, you can have a quality of life, but the quality of life for most people is unfortunately poor.
And short.
And short. So what is it? Well, this is a cartoon. If you look at the right lung, that's a standard lung. The alveoli at the bottom of your deep lung, that's where the oxygen CO2 exchange takes place. And on the right-hand side, you'll see that grey stuff. That's extracellular matrix. That's collagen deposition, which means the lung now cannot transfer the CO2, can't do the CO2 oxygen transfer. So basically, you end up not being able to breathe, and that's IPF. So what are we trying to do? Well, an integrin inhibitor, which hopefully this little cartoon explains it. If you go from the right-hand side, you've got fibrosis. One of the key players in that is a cytokine called TGF-beta, transforming growth factor beta. And TGF-beta is held in an inactive form in the body by this protein called LAP. And in LAP, there's a binding site, and these RGD is amino acid code for arginine glycine aspartic acid. And it's known that alpha V beta 6 binds here. And when it binds, by various processes, TGF-beta comes off and eventually leads to fibrosis. So basically what we want to do is inhibit that process. It's a relatively straightforward hypothesis. Two things to note, that healthy individuals don't express alpha V beta 6, but as the disease progresses, there's more expression, and it's also located in the lung, on the epithelial cells in the lung. So therefore, that's why we wish to treat directly to the lung rather than going through the oral process of absorption, distribution, metabolism, etc. Go straight to the lung. So yes, there are competitors. Supply here was a spin out from UCSF. Their compound is a beta 6, beta 1, and there's good rationale for some beta 1 as well. That's in the lung. Importantly, there's an oral treatment for IPF, and as I mentioned, liver disease. They're looking at PSC also. But for us, they've just had successful phase 2A data. And actually, that's really good because there was 2 other integrin players fell out the market, and people were worried about mechanism of action, but that kind of suggests there's no mechanism of action. There was also Biogen had an antibody, but that was kind of on for a very long time. And what that was doing probably was whacking down TGF-beta to no level. And you need that for other processes in the body. So our inhaled approach is just to tickle it down, but not obliterate it completely. Boehringheimer's got a PDE4, different mechanism, but is going quite well. EMS, I've got this autotaxin process, basically. Interestingly, Alevin Pharma, I've looked at nintedanib and pirfenidone and thought the systemic effects are because it's oral. Can we nebulize it? And that's what they're currently doing. So they've got a phase 1 trial with nebulized nintedanib, and that's going quite well. And again, that proves the concept of nebulizing straight to the lung, which is our approach. So I just quickly go a little bit where we are. Most of the effort this year has been progressing it to the stage of safety. So we've done a kind of single dose study in rats up to 80 mg/kg, no problem. So that allowed us then to go for a repeat dose study, 14 days. We did 2 doses, 10 and 30 mg/kg, and again there was no issues. We have a consultant pathologist who's an ex-colleague from AstraZeneca. He peer-reviewed the slides, and he says what you're seeing is irritation because of the compound was taken with saline, and it was a saline that was affecting the lungs, but there's no drug adverse effects from the drug, which is great news. There's a company in Newcastle called Fibrifine, and they spun out of Newcastle University, and Newcastle University hospitals do roughly 1 lung transplant a month. These guys get those lungs. So again, a pathologist, biopsy confirmed by a pathologist, they take the IPF tissue and cut it up in small slices. In this case, it's a 96 well plate format, and we put our compound in at various doses, and we're looking at markers of fibrosis, collagen 1A1, which is marker of collagen, and TIMP1. And then, interestingly, that those markers were reduced in the wells. But more importantly, we put in nintedanib and pirfenidone with the compound, and we saw an attenuation of effects seen with the 2 standards of care, which is actually really really important because if you did a phase 1 trial, you couldn't take people off standard of care. And so we've shown that we've got an additive effect with standard of care, which is what the tagline will say at the bottom. But
I'm sure it'll come.
You've lost the Midas touch.
oh, there we
Yeah,
go.
there you go.
So no, you've gone the wrong way.
Please take the one. I won't do too many times. I will do them.
So the important thing from our point of view is that yes, it works in vivo. Yes, it's safe. Yes, it works with standard of care. So what do we do next? Well, our development plan next, when the slide comes up, is basically we're now in the position where we want to do safety studies. And so we're today pitching for 2.5 million because that's what we think it's going to take. Because we're going to have to make 2 to 3 kilograms of compound, and we'll do inhalation studies in rodent and non-rodent, probably rats and dog. And inhalation studies are an order of magnitude more difficult than oral studies. But technically, we can do them.
One more.
Yeah, one more. No, 2 more actually. Next one. Right, so we've done all this stuff, we've done all this stuff in green, so we're at milestone 1, apart from, we haven't done a polymorph study, but that's relatively straightforward. So we're now at this position where really what we want to do is do the scale-up chemistry and do the installation safety studies, and we reckon that's about 12 months or so, and then into phase 1A, possibly phase 1B, we might get some human volunteers in that, and our approach would be a nebulizer. Probably not a PDMI, but probably a nebulizer straight to the lung. And that'll be obviously the usual thing with human volunteers, tolerability and efficacy. But we might, with some B people, with phase 1B with patients, we may be able to show some efficacy as well. The standard efficacy is a forced vital capacity, which has to go down, obviously, which usually takes maybe 6 to 8 weeks to come in. But we're looking at some biomarkers, and the FDA at the moment being persuaded that biomarkers might be a good marker. But I can do this now. Okay, so as I mentioned, I was always taught backups. So we have our compound is covered by this patent. It's in the public domain, but we did national phase entry in various countries. It's going through a grant at the moment. In terms of nanomolar, it's, as I say, it's 6. This is the client compound. We had it made and we tested it, and it's 1 nanomolar. We've got 2 backups which are equivalent or better. If you look at the PIC50 numbers, which is a minus log of the IC50, it makes it easier to see. The standard deviation on this screen is about 0.4. So these compounds are significantly more important, are better than our lead compound. But our lead compound, there's nothing wrong with the lead compound in terms of our projected dose demand. But this offers a good backup, just in case there's a problem in efficacy or in toxicology, should I say. And I think one more. That's it. So in summary then, the compound's potent. Physical properties fine for administration to the lung. I didn't say, but the compound is designed such that when it comes back into the systemic circulation, it's cleared rapidly. So there's a very short T1/2 systemically. So we're hoping any systemic effects that you might see with advanced integrin inhibition, we won't see with our compound. I didn't mention it, but there's no off-target effects in a number of things. We've done SIPs, we've done AIMS. So in those there's no problems with SIPs and AIMS either. Sorry, SIPs is cytokine P450s. The repeat dose study, as I say, was good. Proof of concept, mouse bleomycin, you've got to do that. Therapeutic model, 28 days, and our compound was 3 milligrams per kilogram intranasal as a surrogate. And IPF tissue, soluble compound, some backups, and say that's what we're trying to do. I think we're in the position where we can take it to the next stage, and that's our very lean team. We've got Sunil, who's our executive chair, Chris Moody, Alice and John and myself are the founders, and Alice McGowan from NTV in Nottingham University. They help spin out companies, basically.
Thank you.
That's it.
Great. Thanks so much. It's interesting because there's two drugs in the market, they don't work very well. So huge unmet need.
Yeah.
Pliant is basically a Nasdaq-listed company with a multi-billion-dollar valuation. It's the biggest problem with ABB6 has been the target. There's been a lot of failures. People are saying the target has got some effects around the target. You're never going to get a drug.
Yeah.
But if Pliant can show in
Yeah.
phase 2 trial that their drug works well, while the market is very big, they will have an oral, and we think the drug's good as well in our own assays. But we'll have an inhaled version for IPF patients. They take oxygen routinely, so it's a good way to get the drug in. So it's an alternative to the oral. So I think there's a big market. So it's really the bet on this really is is Pliant going to be successful or not? And if they do, the market will heat up very quickly. The last deal that was done with Morphic was like $1 billion of like a few hundred million up front. So it's a very exciting area if Pliant's drug is successful. If it is, we'll probably get hoovered up. So that's the play.
That's where we are. Thank you. Excellent.
Any questions?
So you say it's inhalation.
Yeah.
Where do you think the target is? Deep in the alveoli?
Yes. Yeah.
And have you had some thoughts about how you're going to actually get the drug on the tongue?
GSK have participated in the literature. They've taken it as more right to be long, and they've shown that nebulizer can do that. So that's the better. You're absolutely right. It depends on the droplet size. And I've been speaking to some in the States, some physicists, all about droplet size, and the rate of flow and how long would you nebulize for 10 minutes at X mils per minute. The beauty is, I didn't show the structures, but these dual compounds. So they all had to be drug-like. And so at the end of that, we built up a portfolio of compounds, and we've got one lead asset that were good enough that we decided to spin out a company.